DNA is the genetic material found in all living organisms ranging from unicellular bacteria to multicellular mammals. It is found in the nucleus of eukaryotic cells and in the organelles like chloroplasts and mitochondria. DNA carries information. The question we need to ask is, what is this information for? This information is like a chef's recipe. DNA is our body's recipe for making proteins. The different ingredients we use in these recipes are the 20 amino acids that our body uses to encode protein molecules. You may ask, why does DNA encode proteins and not carbohydrates or lipids? Lipids only have 4 molecules in their largest, most complicated molecule. So we do not need a recipe for them. Triacylglycerides are made up of glycerol and 3 fatty acids only. Some carbohydrates are extremely long, complicated molecule chains, such as in the example of cellulose. But we do not need a recipe for cellulose because it is made up of only one type of molecule, glucose. In a recipe, the order you put your ingredients is vitally important. It is the same case with proteins and amino acids. The amino acid chain must be in exact order, or otherwise the protein chain will not work. Also, for every amino acid position in a protein molecule, there are 20 different amino acids to choose from. Every time we add an amino acid to a growing protein chain, the question would be which one of the 20 amino acids to add. We cannot add an amino acid randomly. Instead, we follow a recipe or a code that guides us and let us know which one of the 20 amino acids should be added to each position. This is the context in which we need to think and understand why DNA codes for proteins, but not any other macromolecule. DNA is the recipe that tells us the order in which amino acids must be chained together to make protein. In conclusion, DNA is the recipe for making proteins. The information carried by DNA is held in the sequences called genes. A gene is a unit of heredity and is a region of DNA that influences a particular characteristic in an organism. Genes only make a small portion of the DNA sequence. As I mentioned earlier, most genes contain the information needed to make functional proteins. These proteins then determine our traits. The trait is a specific characteristic of an individual. For example, their hair color or their blood type. Traits are determined by genes. And remember that genes are the messages in our DNA that define individual characteristics. So the trait is the manifestation of the product of a gene that is coded for by the DNA. It is not which genes you have, but how those genes interact with each other during development that makes you different from another individual of the same species, or from a zebra or a cabbage. The human genome is a complete set of nucleic acid sequences encoded as DNA within the 23 chromosome pairs in cell's nucleus and in a small DNA molecule found within individual mitochondria. Protein coding sequences account for only a very small fraction of the human genome, which is approximately 1.5%. The rest is associated with non-coding RNA genes, regulatory DNA sequences, and sequences for which as yet no function has been determined. The human chromosome length is about 205 centimeters for the male genome and 208 centimeters for the female genome. An individual human cell contains about 6 billion base pairs and contain around 30,000 genes. Approximately 23,000 of these genes are protein coding genes. The number of human protein coding genes is not significantly larger than that of many less complex organisms, such as the roundworm and the fruit fly. Men have fewer genes than women because the Y chromosome is smaller than the X. The X and Y chromosomes are the sex chromosomes that determine the gender of an individual. DNA is composed of nucleotide chains that coil around each other to form a double helix. The two DNA strands are known as polynucleotides as they are composed of simpler monomeric units called nucleotides, just like any other nucleic acids. Each nucleotide is composed of one of the four nitrogen-containing bases: cytosine, guanine, adenine, and thymine, a sugar called deoxyribose, and a phosphate group. The nucleotides are joined to one another to form a double-stranded molecule by hydrogen bonds between the complementary nitrogen-including bases, resulting in a double-stranded structure. The two strands of DNA run in opposite directions to each other and are therefore antiparallel. The nitrogenous bases of the two separate polynucleotide strands are bound together according to base pairing rules: adenine with thymine and cytosine with guanine by hydrogen bonds to make double-stranded DNA. In a DNA double helix, each type of nucleobase on one strand bonds with just one type of nucleobase on the other strand. This is called complementary base pairing, with adenine bonding only to thymine in two hydrogen bonds and cytosine bonding only to guanine in three hydrogen bonds. As a result of this base pairing, all of the information in a DNA double helix is duplicated on each strand, which is vital in DNA replication. This reversible and specific interaction between complementary base pairs is critical for all the functions of DNA in organisms. Thanks for watching.